if you don't mind, contrary to what's up there on that clock, I have about 6:09 or almost 6:10, so if you don't mind, I'm going to get going. I also don't mind, I'm going to be wearing my headphones, and part of the reason I'm going to be doing trying to that the stream is going live, and we'll talk a little bit more about this as we go on. I am also taking a backup audio for you. In other words, I want to give you every opportunity to do as well as you can in this course and to give you as many resources as possible in this course. This lecture itself, as you can see from notes, it's pretty dense. There's a lot to talk about today as well as in Wednesday's class, and when things start to slow down a little bit, I want you to have as many resources as possible throughout this course. And again, we'll talk a little bit more about that as we're going along. Feel free, by the way, to interrupt me at any time. Physiology is something that you should actually be enjoying. Whether you're aware of it or not, you're using physiology, your body, how it works every single day, and your bodies are changing, you're getting older, or you're going to the gym, your muscles are getting bigger. All of this is directly to you, and want you to be thinking about physiology maybe in a different way than you might have thought about it just as a course. Okay, I'm going to go through really quickly how I'd like you to start studying this course to make sure that you're doing well. It is really important for me that you do well in this course. I don't want to just lecturing to you and then just having you not do well on midterms or on your assignments or anything else. I should be here to help you as a resource individual and someone who's going to support you in different types of things related to CBLG600. So I'm going to go through a couple of suggestions. They're just suggestions. You might have your own way to study. You might have your own way to do things. And I certainly don't want to make things more complicated than they have to be. But I do want to talk to you about issues in physiology starting today, right? So how many of you have studied in other courses? Okay, so what can you tell me about the brain that you have learned in other courses? Thanks for your help. Some of you put up your hands, so some of you have learned about the nervous system. What can you tell me about the nervous system? Anything? So if you damage you're in good shape, right? Yeah? No, you're in really bad shape, and we'll talk a little bit about why you're in really bad shape if you damage your brain. What else can you tell me? Do we know everything about the brain? No? In fact, it's the one organ system, and the reason why we actually start this is because it controls so many of the other systems that we're looking at in this course. But the reality is, all the stuff that I'm going to be telling you, the foundational stuff, is really just the surface. We don't know for how you have consciousness. You're aware that you're sitting in this room. That involves your brain. I can't tell you how that works. I can tell you different structures that might be involved, but the brain is really something that we don't understand a lot about. So when we talk about the nervous system, neurons, and how they all interconnect, there's a lot that we still don't know. And I want you to be curious about those things. And I want you to try to engage in that as much as possible. So again, it's completely up to you. If you find that you really need a textbook, go out and buy the 10th edition of the textbook. If you've taken this course before and you have the textbook already, use the edition of the textbook, but you don't have to go out and buy it. Everything that you're going to need to do well in this course, I'm going to be providing for you, and I should be, again, a resource person for you wherever I can. So this is the first summer offering of CBLG600, so I'm going to ask you for lots of feedback, whether things are more difficult or challenging for you and you need a little bit more help, then I'd like to hear that and be talking to you off and on throughout the rest of this course. I do really want you to love physiology. When I was an undergrad student, I didn't like physiology very much, and to be honest, the worst I got was in the nervous system physiology. And I won't tell you the mark I got, but it was between the letter C and E. And it wasn't a very good grade, but it's something that I really, really like now. And I'm hoping that you also really love physiology by the end of the course. So we're going to have a couple of midterms or term tests and a final exam. We're going to have some tutorials along the way using case studies. They're not really going to be material that you should be worried about for the tests and exams. I see that some of you all, other people in this class, that's great. I want you to be working together in small groups to make sure that you're getting prepared in your tutorials for your midterm and everything else. And I'll tell you a little bit more about that in the next few slides. So we're going to have, not this week and not the week after, but just before you write your midterm, I want to have a sort of review session in class where all of you are helping each other to study. We're going to go through some of the material that we've already covered, make sure that you know it, understand it, will do well on the midterm beforehand. I'll be posting some of the rubrics and some of the assignment guidelines for your written assignment that's worth 15% in your course syllabus. And I, again, want you to do really well on that. And I'm going to try to change it up a little bit from how Dr. Agard had set it up. The papers that she has are only related to the nervous system, but I'm going to try to pick a couple of other papers that are maybe outside of the nervous system as well. How many of you like group work? One person, two people, some people aren't sure. I don't know if you guys have the same sort of acronym here. I guess it's AFK. Do you guys have that here too? Away from keyboard, no one's actually answering you back. Your group member who's supposed to be telling you like what's going on, just stop texting you, stop replying your email, and they're away from their keyboard for a long period of time. I don't want that in this class. I don't want group work to be something that's a hindrance to you. I want you to, again, feel comfortable with the groups that you're in, and I want you to finish this course with your sanity intact. I want you to get a good grade. I want you to have learned a lot about physiology, and especially about the topics that we're going to be talking about, but I want you to have this course with your sanity intact, with a good grade, a really firm foundation in physiology. I'm hoping along the way that you, okay, so this week, okay, watch the Raptors. Make sure that you're supporting the Raptors and your playoff run. But I don't want you to be leaving things to the last minute. This course is going to go by really, really fast. 6 hour lectures a week means that you should be staying on top of things over the weekend. One of the things that I'm going to do to help you to do that is to put practice questions similar to what you'll get on your midterm over the weekend for you to practice. They're only going to be there for the weekend. I'll put them back up for your term test, but they're just going to be there to help you study as much as possible. If you guys do a really good job on the 2 tests, I will try to make the final exam not cumulative, unless you want a cumulative final exam. Yes? No? No? Okay. What do you think is a good mark that this group should be aiming for not to have a cumulative final exam? Term test 1, term test 2. What average do you guys think this class is capable of getting? 70? Is that the best that you think of this class? Look around at your classmates. This class is capable of 70. 70? 71. Okay, better. Okay. So let's put it this way. I have high expectations from you. We'll keep it reasonable. If the averages on the first 2 term tests over 72, we'll make the final exam not cumulative. You guys can manage 72, right? 72, oh yeah, okay, good. I like that attitude. So it doesn't have to be totally cumulative. We'll work on those details. We'll see how you do on your 2 term tests, and we'll make sure that you guys are well prepared. Again, some of you don't like group work. Some of you are going to, at one point in time, be applying to different programs, whether it's professional school or whether it's for graduate school or research. Maybe it's even in volunteer work, whatever it is. Nowadays, when I get a request to do a reference letter, the reference letters often ask for all these different types of things, like what's the motivation for graduate school. So for example, I usually write, it's really good, it's a superior A+. But there are certain things, unless you do group work, I can't actually answer with all honesty. And this is a typical sort of professional school / school application, your ability to work with other people. I don't know. Unless I see you working in a group, I can't really respond to that, and I'd like to be able to. Ability to organize other people. If your group members aren't responding back to you, what are you going to do? Right? Are you going to let me know about it and try to help you? What all of this is really saying is that I want you to think of these different assignments not as a way for you to get lower marks or to make your life difficult, but for me to be able to engage you and to be able to help you down the road, whether you're applying to graduate school or to medical school or wherever you're applying, because these are the types of questions that I get asked when I'm asked to evaluate you after this course is done. So I'm hoping that you're not looking at it as, why do I have to do this? These are some of the reasons why I want to see your abilities in these different types of things. Since we're starting with the nervous system, one of the things that the nervous system is really, really good at is its ability to maintain what's known as homeostasis. Your ability to walk, for example, a feed forward and a feedback system. You can't walk unless you have feed forward and feedback mechanisms. We're going to be talking about those, but these feed forward mechanisms tell you, like, as long as this ground is level, then I'm okay. I'm able to just keep walking the way that I normally do. And it tells you that based on previous experience, how you should be studying for this course, for example, how you should be getting ready. Sometimes, though, we have these feedback mechanisms. Something comes up, something doesn't go right for you, and then you need to be able to adapt. And these feedback mechanisms are really important as well. In order for you to be able to do both of these, because right now you're in this really unstable time. Like, I've never had this prof before. I've never been at Ryerson before. I have no idea what this course is going to mean to me. This is a really scary thing. This is the beginning of the course. It's an unstable time. And you're going to have to adapt really quickly, use your best judgment and things that you know work well for you. And some of those things that I can help you with is, if you can, in terms of the feedback mechanism, for every hour that we cover in this course, so we're in the first hour, we'll get into some slides soon, I want you to use what we call a 3x3 mechanism. So for every hour in this class, just take one concept that I'm completely lost. When you started talking about the action potential in the third hour, you lost completely. I need to come and talk to you. So that's one of the three. The second one is maybe just pick out something that is really important or exciting or interesting to you and use that sort of as a frame of reference. I can't remember everything from the lecture. The reality is, as soon as you walk out that door today, within the first 15 minutes, you will have forgotten 60% of the lecture. But you'll be able to hang on to at least one thing from every hour. That might be the anchor you need. Even if you don't do 1, 2, the third thing you should do is, in lecture, you should be able to identify in every hour 3 different slides that you think might be on the final exam, because you might be using this feed-forward mechanism. You know, I've been in other courses in physiology, and I think that one of the things that he's probably going to be asking us about is this slide here. So pick 3 different slides that you think might be on this on your midterm and everything else. So I'm your prof, you're my students. My job is to help you to understand the material, try to get you excited about this material, and then also to really help you to do well. I need your feedback and ideas about how to make you, or how to get you to do well in this course. So before we get into the rest of today's material, do you guys have any suggestions? How can I help you based on other courses or based on your own experience? How can I help you to do really well? No suggestions? Nothing? Yeah.
Like, just give us a hint, or just let us know that, okay, this whatever slide is important.
Okay.
Like, focus
Done. Okay, done. Good suggestion, already done. Next. Any other suggestions? Give us an idea of the slides that are important for the test. Yes? Yep, we will be doing that in groups. Yep. Any other questions, comments, suggestions, ideas? Nothing. If you think of anything throughout today's lecture where you want to just chat about it quietly, then let me know. I'll be happy to try to entertain whatever ideas that you have. So we're going to be starting with the nervous system. We're going to be looking at some of the different cell types that are involved in the nervous system. How many neurons do you have in your brain and spinal cord, by the way? Not important for your final or midterm or anything else. How many do you have? You have 2 cells? Hopefully not, unless you're Simpson. You should have lots more than 2 cells. How many do all of you have in your 20s? How many neurons? Any idea? Guess. Million, 2 million, 1 billion, 2 billion? A lot? Okay, I like that answer. It's not a multiple choice test question answer, but so you have a choice of 10 million, 100 million, 60 billion, or 1 trillion. 60 billion? Oh, you guys are a good class. Good at guessing. Okay, nice. So you have somewhere between 60 to about 100 billion neurons. It's obviously really hard to count. The best estimate is that you have about 60 to 100 billion neurons within your brain. And this is the structure that we're going to be talking about. We'll be looking at some of the anatomy, some of the structures, some of the cellular components, and then we'll talk about some of the functional concepts. So as I mentioned to you earlier, and again, you don't have to worry about trying to memorize this slide. I'm going to tell you the slides that you should be concentrating on, that you should be worried about for your term test. I want you to try learning. I don't want you to try like memorizing everything on the spot. Being a electronic recorder where you're trying to type every single last word down, I'm going to tell you what you need to know because I want you to actually learn something rather than trying to memorize as you're going along. So the nervous system is really important. First off, whether you think so or not, it's involved in your ability to maintain homeostasis. And again, I want to hear a little bit about what you know from your other courses. If the whole brain is involved in homeostasis, there's one part of the brain that's really, really important in homeostasis. Do you know that one part of the brain? One structure. Can you tell me what that is? It starts with H. Hypothalamus. So we'll talk about the hypothalamus. Again, you don't have to worry about it right now, but it's really important for maintaining your homeostasis, your temperature, the way in which your fluids are regulated, the amount of fluid that you have in your body. All of that is regulated by things like the hypothalamus, and it's there to coordinate physiology as well. Without your brain, you really couldn't process the different signals that are coming in, and it's there to help to monitor both signals from inside your body as well as external stimuli. And we're going to talk about this not today, but in Wednesday's lecture. So we're going to start talking a little bit about how your brain integrates information. So you're able to see things, feel things, and do everything that your 5 senses tells you is going on. But without your brain to process this, and without your brain working properly, and it requires an active circuit, and the circuit has to be intact, that you're not going to be able to integrate information. Because this is physiology, and because it's a real world type of physiology, can you give me an example where integrating information in your brain might not work? Any ideas? So when might something go wrong where your brain, spinal cord can't process sensory information and can't, as a result, initiate the appropriate response? Can you think of a situation? So this happened to me about 2 months ago. So 2 months ago, I was walking up the stairs on the TTC, and as I'm walking up the stairs, my right arm and my right leg stopped working. So it suddenly, I didn't know where it was coming from. I couldn't see really well out of my right eye. And this is where, for example, knowing a little bit of physiology actually was really important to me. I had what was known as a transient ischemic attack. I didn't have a stroke, in other words, but it was pretty close to it. For about 5 minutes, my vision was really affected. My brain couldn't process the signals, but it's just the right side of my body. And I knew right away that I was having some sort of ischemic attack. And this is why your brain is so important. It is integrating that information. It is allowing you to have that appropriate initiation of response. I tried putting my foot up to get on the stair, but it just wouldn't listen. And it's part of the reason why I'm so interested in the nervous system, because it helps to initiate responses, things like controlling your muscles as well as your glands, and of course allowing you to think, do conscious processing, to have your emotional responses, all of that is regulated by your brain. So I'm going to start taking you through different parts of the nervous system, including your brain, the next few slides, and we'll talk about the brain, spinal cord, how all of that sensory information gets into your body. We'll talk about the subdivisions of the nervous system. There are really two parts to your nervous system. It's not just your brain and spinal cord. You have all this other stuff out here that leads from your arms and legs to your spinal cord, and that's the peripheral nervous system that involves sensory receptors, that are in your fingertips, on your arms, etc., and nerves that convey them back into the CNS. So again, if we take a look, we have two different parts. The brain and spinal cord is traditionally anatomically considered to be the central nervous system. It integrates all this information. Remember, integration of that sensory signal. But in order for us to actually get sensory signals into the CNS, your brain and spinal cord, we need the PNS, or the peripheral nervous system. And the peripheral nervous system includes all of the other aspects that we'll go into in some more detail in the upcoming lectures, and starting next week, for example, on Monday. So there is information coming from your fingertips actually won't know what you're pressing up against. So there's a sensory receptor, there's a nerve that's transmitting that signal. It's getting up into my spinal cord, and my brain, without me looking, is able to tell me what I'm pressing up against. Okay, so this is where we need these two different divisions of the CNS and PNS. Is that okay for all of you? Hopefully it's a really cursory overview. You don't have to worry about the details, just to give you an idea of the different types of things that we're going to be talking about. So before I get going, do you guys know what Bell's palsy is? So you guys are really, really young, right? So I mean, that's not a trick question. You're either going to say yes or you're going to say no. You're really young, right? Yes. Okay, thank you. Do you guys remember a prime minister by the name of Jean Chrétien? Yes, no, no? Oh, boy, you guys really are young. So Jean Chrétien actually had a viral infection, and half of his face, the facial nerve, was actually affected, and he had Bell's palsy, so he couldn't really, it looked like part of his face was actually not responsive. His muscles on half of his face didn't work. And this is because of the way in which our nervous system, our peripheral nervous system, is organized. So again, we'll talk more about sensory receptors, not today. We'll talk about it on Wednesday, and I'm again just giving you an overview. You don't have to have memorized anything right now, but we need to understand how to get these sensory signals from the free nerve endings related to pain and all of that in toward the CNS before we can actually understand what's going on. So these sensory receptors help tell us if we're seeing something, if we're looking at something that's a light source. It tells us about what sounds we're hearing, what we're smelling, temperature, pain, touch. All these sensory systems are working through the PNS. And they're traveling from the sensory receptors in your fingers, your eyes, etc., through a nerve bundle of axons that I'll get to in a few moments that take it into the CNS. Remember, the PNS transmits information into the CNS, and we say that it works through a nerve. So think a little bit about this, because again, I know you guys are really bright. We have two different aspects of the cranial nerves, and then we have spinal nerves. So the cranial nerves originate from the brain. There are 12 pairs of them. So there's cranial nerve number 1, number 2, number 3, all the way down to cranial nerve number 12, which controls how your tongue moves. Why do you think we have the cranial nerve, and then we have another set, the spinal nerves, where we have 1 pairs? Why do we have 2 different? Any ideas? So one originates in the spinal cord, the other one is up in. What do you think is different about them? Yeah?
A lot of senses are located in our face, so I think there's kind of related to the upper.
Exactly. So, right, you have it exactly right. So the cranial nerves innervate, tell us about sensations everywhere above the spinal cord. Your head region, in other words, about your neck. When you're tapping on your face, you're activating cranial nerves. And then spinal nerves are everything below. So your body, for example, is innervated by your spinal nerves. And that's again, great. So we'll talk more on Wednesday about ganglions or ganglia, which are collections of neural cell bodies that are found outside of the nervous system, the CNS or the central nervous system, and then we'll talk about plexi or groups of axons as well. And it's just, again, an overview about the complexity of the anatomy. You don't have to memorize anything from it. It's just to give you an idea of how information gets conveyed and some of the anatomical terms that will be covered. Do you guys need anything cleared up on the slide before we go on? Yes?
How far above the spinal cord are we talking about? Just that.
Sorry? Just the...
The cranial nerves,
Yeah.
how far do they control above?
So how far above it? When we get into the neuroanatomy, so it's not the spinal cord stops and then the brain starts, right? It's actually continuous. And the medulla, which is the last part of the brainstem, brain, it's continuous with the spinal cord. So generally speaking, it's everything above your neck. Okay. Any other questions? Happy to answer them. These are good questions and hopefully things that interest you. So this is a slide that I'd like you to make sure that you know. There are a couple of details on this slide, and every sort of course and every sort of discipline has its own language, right? And have all of you taken an anatomy course before in your first or second year? No? Yes? Some of you have, some of you haven't. So I'm going to tell you the most important parts of this, because if you don't understand it now, you're going to have a harder time understanding it on Wednesday and again next Monday. So I told you that the peripheral nervous system takes signals into your spinal cord, into your brain. It's the sort of conduit from out here into your spinal cord and then out your brain. But there's a very, very organized way in which it does this. It's not random. In other words, we are really talking about your brain as a wired system, and the wiring is very, very precise. So when we are talking about signals, sensory signals that are coming back into your spinal cord, the central nervous system, up into your brain. So when I'm tapping on this whiteboard over here, the sensory receptors are sending signals along the nerves back into my spine and then eventually up into the brain. Without me even looking, I can tell you that I'm not touching a whiteboard here. I don't need my vision or anything else. My sense of touch is enough to do that. So there's a couple of things I want you to know. When we talk about information going into the spinal cord, into the CNS, we talk about it being an afferent signal. Okay, it goes in toward the CNS. That information that is going into the CNS, and here is part of the CNS, this is the spinal cord, the part that is going in, that sensory information, it doesn't matter if it's coming from my leg, so down lower in the spinal cord, or my arm, all of that information, sensory information, always comes in to the spinal cord through what is known as the dorsal root. So it always comes in up here, in other words, okay? So two things I wanted you to remember. Sensory information is an afferent signal. It goes into the spinal cord. And then when we talk about information coming in, those nerves from the PNS, those nerves send their axons in through the dorsal root of the spinal cord. Is that okay for everyone? Anything I can clear up for you, not 100% sure about? Okay, so the same spinal cord, so if we're taking a look at it, the sensory part is up here in the dorsal region. It receives afferent signals about what's going on in the outside world. And then the other part of the spinal cord is found in this region, which is known as the ventral root of the spinal cord. And this ventral root is an efferent signal. So imagine this, right? So I've just touched something, but need to move my hand away really quickly. I just touched the stove and it's really hot. The sensory signal, the hot signal, is an afferent. It's stimulating my sensory nerves, but the movement away, the efferent signal, is controlling my muscles. That's all being initiated at the level of the ventral root. moving a muscle, in other words, requires nerves that are found in the ventral root to become activated. Okay, is that okay? About efferent signals, where something leaves the nervous system and controls muscle movement, then we're talking about the ventral horn or the ventral root. And when we talk about input of signals, then we're talking about the dorsal root. Any questions? Are you guys all okay with this? So in about 2.5 to 3 weeks time, because again, it's really, really compressed, if I ask you a question related to the dorsal root or sensory signals coming in or controlling muscle movement, you guys are going to get 100%, every last one of you, right? Okay, I will hold you to that. Remember, our goal is to get 72 or higher. Did you have a question?
I was just wondering,
Yeah.
the ganglion part, how is it different from the spinal nerve part?
How is the ganglion different? Okay, so we're going to talk about this. Nerves are different. Like they have different shapes. And the way that this works is the cell body actually sits out here in a collection known as the dorsal root ganglion, but it sends one projection this way and another projection this way. And the cell bodies themselves for this peripheral nerve signal, because this is peripheral nerve, it's outside the spinal cord, right? Do you agree it's outside the spinal cord? If this is the spinal cord, this is outside of it. This is a pseudo bipolar cell or pseudo bipolar neuron. Was there another question over here? And again, you don't need to worry about those details. Another question? No? Okay, so that's one you can kind of put in the bank for your upcoming term test. You should know the difference between afferent and efferent, and what efferents control are muscles, and they're found in the ventral horn or the ventral root, and then sensory signals come in through the dorsal root at the top. Okay. All good? I will actually hold your class to 100%. If anyone doesn't get 100%, I'll be knocking on your door late at night asking you to go over this in your notes with me. Okay. I'm not going to spend a lot of time on this right now. And again, it's just to give you an overview. We're going to start talking about the somatic nervous system and the autonomic system. We're going to talk about the somatic in more detail in the next class. But if we're talking about the movement of muscles through the ventral root, then one thing you know is that you can control consciously. Like I see something over here, I want to pick it up. That's voluntary control. And that usually involves one neuron being activated. It's something that involves a synapse between a nerve and a muscle, for example. And we'll go into more detail later on. The only thing that I'll ask you to remember, and you don't have to memorize it right now, is the somatic nervous system. When we talk about voluntary movement, it's all skeletal muscle. You can control skeletal muscle. Unless you're having a stroke, unless you're having a seizure, you are always in control over your skeletal muscle. Okay? What other muscle types, as physiology students, are you aware of? Heart? Okay, so we're sophisticated in this class, so we don't talk about heart muscle. We call it something else. Cardiac muscle, and then what's the other muscle type? Smooth muscle. So can you control your cardiac muscle? No? Can you control your smooth muscle? Are you sure? How are you sure? Okay. Let's try an experiment. I don't know if you're hungry. Let's give it a shot. I'm using my skeletal muscles now to open this up. Someone somewhat hungry would like a little chocolate? 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 Yeah, you can take one. Yeah, I'm sure you can take one. Chocolate? Chocolate? Yeah, take one. Anyone else? Why don't we pass down this way? I think there's a couple of volunteers down that way. All right. So whenever you're ready, go ahead and start eating. And as you're eating, and it's really small chocolate, I have to apologize. as you're eating, we'll go over the autonomic nervous system. And this is where the CNS doesn't make a synapse or connection to skeletal muscle. Here we're talking about a neuron from the CNS making a connection to smooth muscle or to cardiac muscle and even to some glands we'll talk about a little bit later on. We are not able to control those. It is not under our conscious control. Our conscious control means that if I want to stop walking, I stop walking. So for those of you who have started eating and have swallowed the chocolate, I'd like you to stop digesting that chocolate right now. You can't, right? And this is the difference between skeletal, smooth, and cardiac. Some of these things are under our control and some are not. We are going to come back and talk about these different autonomic divisions of the PNS in terms of how the sympathetic nervous system controls different parts of your body getting ready for physical activity, or the parasympathetic as well for vegetative functions, and the enteric system that controls how your GI tract, the digestive system, gets regulated as well. We're going to come back to all of those just to give you an overview of the different variations of the PNS. And again, so far, the only thing that you needed to worry about is the afferent, efferent, ventral, dorsal connection. Any questions on this before I go on? Because you're about to get into the next slide that you need to memorize for your term test. Any questions? If you're wondering if I have more Twix, I do. So we'll do that maybe after the break. So take a look at this red neuron here, which is part of the autonomic nervous system. Okay? If I was trying to control the skeletal muscle system, where would this nerve be sitting? If I wanted voluntary control, where would it be sitting? So we just learned this, right? Dorsal, but I want to make a muscle move. So I would have it down here in the ventral region. But take a look at what is happening here. This autonomic nervous system isn't sort of way up here, the dorsal. It's not receiving sensory information, and it's not controlling skeletal muscle, so it's not down in the ventral region either. So the autonomic nervous system is actually found right in the middle. It's found in the intermediate zone. It's found at the level of the intermediate region between the dorsal and the ventral region, okay? And all of these things are not involving the control of the contraction of skeletal muscle, but of the movement of some of those other muscle types, whether it's the heart, whether it is smooth muscle that we find in the large intestine. This is the autonomic nervous system. This is where we find neurons that control the movement or contraction of those things. And this intermediate region, or this intermediate zone, is something that I do want you to make sure that you know, okay? So there is a precise order, in other words, where we have in the spinal cord, which is part of the CNS, where we receive signals. That's all done here at the top. We will be able to cause the movement of skeletal muscle down here in the ventral region. And then talking about the control of smooth muscle, there is a precise region anatomically in the CNS that's responsible for contractions of things like the large intestine or other smooth muscle. So again, this gets back into the precise wiring and the precise nature of how the nervous system is put together. Any questions on this before I go on? Yeah. Not right now, no. When we get to the autonomic nervous system, you will, but for the time being, I just want you to have an idea about the way in which the spinal cord as part of the CNS is put together. There is a topographical order to this, in other words. So again, you don't have to memorize this, but we use our receptors, and then they signal through the sensory nervous system. It travels up into the CNS through both the spinal cord and then into the brain, and then it will send another signal back as appropriate through the motor nervous system to cause effectors to move, whether it is an effector like skeletal muscle or others. Yeah, do you have a question?
Could you please go back to the last slide and go over what you said?
What did I say?
From
The spinal cord from top to bottom. Okay. So we know from the last 2 slides that we get sensory information coming in at the dorsal root of the spinal cord. And again, the CNS includes the spinal cord, and it's receiving this peripheral input at the level of the dorsal root. We know that if we're controlling the movement of skeletal muscle, it's down here at the ventral region or the ventral horn of the spinal cord, and that will cause skeletal muscles to contract. But when we're looking at the control of smooth muscle contraction, things like large intestine, which is a smooth muscle, then we find those neurons at the level of the intermediate zone. Okay, is that okay? Any other questions? Okay. So I told you at the outset that when we look at just the neurons in your spinal cord has about 1 billion neurons. Your brain has about 60 billion to 100 billion neurons. But if we take a look at the nervous system, you actually have a number of other cell types. It's not just neurons. Your brain has a number of other cells. Your brain has glial cells, as well as the neurons that we will be talking about in more detail. Okay, and we'll talk about some of the organization of the neurons in the next few slides. And again, have all of you had at least one lecture on the nervous system and neurons before? Yes, so it's all right? Okay, we'll see in a second. So I heard at Ryerson you guys actually had a concert going on the weekend that had Drake. Were any of you at that concert? One? Okay, that's okay. One person. We'll try to add you on Facebook later and try to figure out the different pictures that you took at the concert and everything else. If we were to break down the neuron, whatever type of neuron that we're looking at, the neurons, generally speaking, have 3 different sections to them. And these sections are one that I want you to be able to tell me a little bit about. And whenever you see Drake, because he's everywhere these days, whenever you see Drake, you should be concentrating a little bit more on this slide, because you'll likely see something from this slide on your term test. Okay, so this is a slide, in other words, when you're going back on the weekend, you guys also had the weekend over here at Ryerson at some point too, right? No? Last year, okay. So when you're going back on the weekend to review, this is a slide that you should spend some time on. When we look at the neural cell body up here, we find a number of different things in this particular compartment. Again, a neuron, generally speaking, doesn't matter what neuron you're looking at, has a cell body with these dendrites associated with it. Then we have an axon that's normally associated with it. And these 3 different parts are what make up most of the neurons that we'll be looking at. So when we look at the first part, the neuronal cell body, we will often see the nucleus in here, and then we'll also see what is known as Nissl substance. Have any of you taken a histology course? Have any of you ever heard of Nissl staining before? No? In neuroscience and in neurobiology, Nissl substance is actually really important. It can tell you if you are missing neurons, if your neurons are healthy, if there's any damage, gone stroke or other injury. This Nissl substance is really chromatophilic means. It likes light, almost close. It likes color, that's right. It takes up staining, in other words. These little Nissl bodies that you see throughout, these little sort of yellowish lines that you see throughout, they like to pick up dye, in other words. They like to pick up color. When you stain a neuron, this Nissl substance is the one that is being stained. And part of that is because it is where proteins are being synthesized. So we see protein synthesis in the rough ER that gets stained. Lots of it, in other words, at the cell body, cell types. And then we'll talk about these dendrites in some more detail. And then we'll talk more about the axons. And again, you don't have to worry about all of the different terms that you see here. I'm going to go through each of them in turn. So for the time being, the most important thing that you should know from this slide are the 3 different compartments that make up a neuron. What Nissl substance is actually representing in this first bullet point, and then I'm going to come back and talk to you about all of these other points as we get into the action potential in a few slides. questions about this or anything that you want to talk about right now before we get going? No? Okay, so this again is a another sort of slide that I'd like you to make sure that you are aware of. I don't recall if this is actually on your slides directly. Did I write retrograde and anterograde on your slides? Okay, so one of the things that happens is you guys have learned about other cell types in other courses, right? What other cell types do you know? Sorry? Epithelial cells, yeah, okay, epithelial cells, great. What's special about epithelial cells? They line everything, that's 100% correct. Internally, outside of epithelial cells as well. What else do you know about these cells? So relatively speaking, some of them are a little taller, they could be columnar, they could be cuboidal, they could be squamous, whatever it is, they're relatively short. Neurons can be incredibly long. So in theory, like one neuron, the cell body could be sitting up here in my brain. Axon could be about 1 meter long, and that's a very long distance, right? Where did I tell you the proteins get synthesized? Where's that chromatophilic substance found? Rough ER, what compartment is that found in? So it's near the nucleus, it's up in the cell body, right, in the soma. So the soma is way up here. The axon is sending its projections like 1 meter away. Do cells need proteins? They do. And so they need to be able to transport this. And this axoplasm, sort of the fluid that's found inside the axon, moves from the cell body down toward the terminals. And it's important because we need to move different proteins that are formed in the soma or the cell body, things like cytoskeletal proteins. We need to move organelles from the cell body all the way down to the terminals, and sometimes that's a very, very long distance for them to go. So you have a choice here. You have two different types of movement. So there is retrograde movement and anterograde movement. What do you think represents for bullet point? It's anterograde. So we talk about the movement away from the cell body as being anterograde. And this is the form of anterograde movement at the top. There's also another form which is retrograde. And sometimes what happens is you need to move things back toward the cell body. Things like different damaged organelles, in order for you to be able to process, get rid of those damaged organelles at the periphery, like down near the synaptic terminals, you need to be able to move them back to the cell body before they can be eliminated. You might have to recycle plasma membrane from like 1 meter away, and all of that requires this retrograde axoplasmic transport, and all of this is moving back to the cell body. Think a little bit about this. You guys have heard of at least rabies, right? Yes? What happens in rabies? Let's not to be worried about. Let's just go around downtown. That's a nice looking dog over there. Let's go pet him or her. Yeah?
Like your vision, you start seeing like different things, so you become really sensitive to light.
Sure. So one of the things that we know is that your brain can get affected by different viruses. In fact, your nerves are actually a great place for viruses to infect. So herpes virus, for example, you might think of it sort of as a skin lesion, but in fact, it's really a nervous system lesion. HIV is exactly the same. HIV loves to hide out in the CNS. But for them to get there, they have to actually travel back along a nerve. So think a little bit about this. A dog who has rabies has just bitten your hand. That hand that's been bitten, some of the virus that's found in the saliva is being taken up by the site of injury. It gets transported back along the nerve in a retrograde fashion back to the cell body. This is the way that viruses like to enter into your nervous system through those different types of routes. Okay, so again, this is a slide that I'd like you to make sure that you spend some time, go over on the weekend if you can, and it's something that you should know about retrograde transport, anterograde transport, and the different examples that might be associated with it. Any questions on that at all? So I had asked your colleagues before the class actually began if we should take one break or one big break of 20 minutes or two smaller breaks of 10 minutes. They agreed that we should take two smaller breaks of 10 minutes. So I think now is actually a good time to take a 10-minute break. Okay, and if you have any questions, come in, chat with me, and I'll be happy to try to answer them.
Sort of like back to you.
So just like having which right now, next few people that enter this classroom, you know if they're the one in this classroom. No. It's the same thing. Your service, your rate of entry is achievable. In fact, your nervous system is actually built and has receptors like the GP120 receptors found on your neurons that allows HIV to actually get into those receptors and then be transmitted.
What about the injections? Like I only heard about the injections. I've never having injections for something like that. Yeah. Is it like this? Does it like follow the same pathway? It's like retrograde motion and then goes.
It's different. You'll get
Don't you? Amazing. No, I'm from U of T. That's why. Environmental biology and Spanish and French. So I'm new to Ryerson and everything. So I was wondering where this live is because I used the online system to upload previously. For
usually they try to do
yeah, I got you.
you taken to it could be. So this portal is you can either go here, but if you watch what I'll tell you, it's called D2.
Today on my that have been so. Did you upload the slides like today? Oh, okay. Oh.
So you go down to,
You click whatever, yeah.
if you go into content,
Mhm.
then you can see the
Okay, fantastic. Awesome, thank you.
It's a little different than Blackboard.
I know, Blackboard's not awesome. Like, you know.
It's also weird on the Facebook side as well. So like the
Is there a group or thing?
yeah. So all of the material, all the work, everything's weird.
Okay.
No, it's not me. It's just. Okay.
So, member also be yeah,
Even if you're going to be on painkillers.
yeah, yeah.
I'd say it's up to you whether you stay in the first part. It's just if you do stay in the first
Yeah. That'd be very ideal. Of course. Of course. Listen. Listen. Well,
If you don't mind, we still have a lot to cover. I want to make sure that we cover as much as we can today so that we're not falling too far behind, and that over the weekend you have an opportunity to go back and review these slides. And again, if you, I know it's really, really hard to do that on the weekend. Some of you might be doing, but if you can review, even if it's only like 1 hour for each of the lectures that we're doing, like just spend 1 hour going back over the slides, you can do a lot to help yourselves do well on the term test and everything else. Some really good questions related to the different types of neurons. We have this are known as interneurons or association neurons, and they connect between different groups of neurons. They might be connecting different sensory neurons, etc. Another way to look at them, though, is structurally. So they not just differently, but they will microscope if you stain them. If you were to stain neurons, by the way, what would you stain them with? Or what would if I microscope? What is a substance you just learned of? Yes. So you might want to stain for Nissl substance. And if you did stain for Nissl substance, you might be able to see, for example, a multipolar cell, a multipolar neuron. This is bipolar because it's just one process this way, another process this way, and so it looks more like it's bipolar. We say this is multipolar because it has a number of branches going off, and this sort of pseudo unipolar, it looks like it has one process, but because the cell body is sitting over here, it actually isn't unipolar. But again, the most important things are not what is the shape that you see over here. It includes things like this. So when we look at classification, it turns out that when we look at the brain, the vast majority of them are multipolar. They look a lot like this. They don't look as much like this. They look a lot more like this type over here. And these are also the motor neurons that we looked at earlier. So motor neurons up here that send, which are efferent types of peripheral neurons that send action potentials away from the CNS toward muscle to cause them to contract. Most of them are these multipolar type. The bipolar ones are ones that we're going to see a lot more of on Wednesday that are involved in the retina, in the eye. We also see them in the olfactory system, in the nose. And so these are more involved and more likely to be found in the sensory systems that we looked at. So motor neurons, sensory neurons, and these unipolar ones are ones that we will also see in the sensory system. So the way I like asking multiple choice test, example of weekend, just review it, might be something like this. It might be a brand new type of neuron in the spinal cord, a multipolar neuron. Which of the about Chris's neuron might be correct? And then you'll have a list of things. It is most likely a sensory neuron. Probably not, right? Most likely toward know that